This is the 2018 AQA AS Biology Paper 1. Before we start, I just want to make some general comments that were in the examiner's report about this paper. So the mean mark for this paper when it was sat in 2018 was just over 30 marks out of 75. The major general comments were that students found it difficult to express their ideas in clear, concise English. They ignored some of the instructions that were given in the questions, but I'll point some of those out as we go through the questions. And that a lot of math skills were lacking. Students really struggled with some of the maths questions. So again, that's something to work on before you go into the exam. So this first question concerns the blood system around the human body. And you're asked to identify certain vessels. So the first one is the aorta. So the aorta is the artery that goes from the heart to the rest of the body, which is going to be this one here. So the answer to the first part is D. The second one is the renal vein. So anything to do with renal is going to be the kidneys, and the vein is the vessel that moves away from the organ. So in this case, that's going to be this vessel here, which is G. Finally, for this part of the question is the vena cava, and that is the artery that goes back to the heart, having been all around the body, which is this one here. So that is going to be F. Okay, part 2 says name the blood vessel that carries blood to the heart muscle, and we've got that in bold. So this isn't asking for a vessel that's taking blood back to the heart. These are the vessels that are feeding the heart muscles, providing it with oxygen and glucose. So that is going to be the coronary. So again, coronary is anything to do with the heart, and it's going to be arteries. Okay, part 3. You're given this photograph of a mitochondrion and you're asked to produce a scientific drawing of the mitochondria in figure 2. Okay, it says label the following parts. So you've got the matrix and the crista. Now, again, looking at the examiner's report, this question's worth 4 marks, but only 2.4% of students got the full 4 marks. So a lot of students fell down. It says draw a scientific drawing of the mitochondria in figure 2. So it wants an accurate representation of this. So similar size, similar number of crista that are coming out in the correct direction. So you aren't just to draw a general mitochondria, it's this one that you've got to draw, and if you don't, you're going to lose marks. Students also fell down on the labels, so this space in between is the matrix, and these folds that are coming out of the membrane are the crista. Remember, it's got to be a similar size, and put your magnification scale on there. Also, it asks for a drawing. A lot of students lost marks because they produced a sketch. So if it was a drawing, you want to draw something like that in a single line, where sketch, people are doing this. You're not going to get the mark if you do a sketch. It has to be a drawing. Some of the advice that the examiners give is use a sharp pencil so you can actually represent the fine detail. Don't shade it and don't use any colours. It's just asking for a simple pencil drawing. Okay, and if you do any of the things like the sketching, the shading, you're going to end up losing marks. Question 2. What is a monomer? So some students put for this about building blocks. So we want to be a bit more precise at A level about our definitions. So even though it's only worth 1 mark, there's 2 bits that this answer requires. So we need a repeating unit, which larger molecules are made from. So you need the idea that it's part of a larger molecule and it's the monomer is this repeated to make that larger molecule. It says lactulose is a disaccharide formed from 1 molecule of galactose and 1 molecule of fructose. Other than being disaccharides, give 1 similarity and 1 difference between the structures of lactulose and lactose. Now, when you're answering a question like this, where it says difference, you've got not just say what a difference is, you've got to explain how it's different to the other thing. So similarity is that they both contain galactose, but the difference is that lactulose contains fructose. Now some students just left the answer at that and they didn't get the mark because you're not saying how it's different to the other molecule. So you've actually got to explain that. So in this case you'd say, but lactose contains glucose. So remember, if it's a difference, you explain there's 2 parts to it. You should explain explicitly how it's different. So next part, following digestion and absorption of food, the undigested remains are processed to form feces. The feces of people with constipation are dry and hard. Constipation can be treated by drinking lactulose. Lactulose is soluble but it's not digested or absorbed in the human intestine. Use your knowledge of water potential. So we're going to have to mention what water potential is to suggest why lactulose can be used to help people suffering constipation. Now obviously water potential is to do with osmosis of water. Now what you've got to remember is water, the movement of water through osmosis goes from an area of high water potential to a lower one, okay, making the water travel to those lower water potential Question 2. What is a monomer? This answer, even though it's only worth 1 mark, requires 2 bits. So it's a repeating unit which forms large molecules. So you've got to get over the idea that it's large molecules that are made up of these repeating units. Some students used answers like building blocks. At A level, we're expected to use more sophisticated language and definitions. So in this case, saying that we've got a repeating unit from which larger molecules are made is a much better way to phrase it. Lactulose is a disaccharide formed from one molecule of galactose and one molecule of fructose. Other than being a disaccharide, give one similarity and one difference between the structures of this and lactose. So the similarity is they both contain galactose. So that's fairly straightforward. So both contain galactose. Now, with the difference, when you've got a answer that requires a difference, you've actually got to say how it differs, what it is about the other thing as well that's different. So lots of students wrote lactulose contains fructose. Now, lots of students left it at that. Now, you wouldn't get the mark because all you're saying is lactulose contains fructose. You're not saying how it's different to the other molecule. So to get the mark, you've got to put but lactose contains glucose. So part 3, following digestion, the absorption of food, the undigested remains are processed to form feces. The feces of people with constipation are dry and hard. This can be treated by drinking lactulose. Lactulose is soluble, but it's not digested or absorbed in the human intestine. Use your knowledge of water potential to suggest why lactulose can be used to help people suffering constipation. So obviously with the question talking about water potential, we're going to be talking about osmosis. So remember, osmosis moves from an area of high water potential to a low one, and by adding a solute to a solution, that lowers the water potential of that solution, i.e. it makes it more negative. Okay, so what we're doing by having lactulose there is lactulose lowers the water potential. Okay, so by having that lactulose in there, you're adding a solute, so you're making the water potential more negative. So making the water lowers the water potential of the contents of the intestine. Okay, so what that does, because you've now got a low water potential, you're going to attract water into it. So for the next part of the answer, so water enters due to osmosis, and therefore softens the feces. So by water going in to the intestine, that's going to go into the feces and therefore make it softer. Now, during the exam, some students got confused about lower and high water potentials and which direction the water moved during osmosis, and by adding a solute, whether that lowers or higher water potential. So just remember, by adding a solute, then that lowers the water potential, which makes it more negative, and osmosis makes water go from high water potential to a lower one. Okay, lactulose can also be used to treat people who have too high a concentration of hydrogen ions. The normal range of hydrogen ion concentration is this. A patient is found to have a blood hydrogen ion concentration of this. So note that this is higher than this because we've got 10 to the minus 7 rather than 10 to the minus 8. Calculate the minimum percentage decrease required to bring the patient's H+ concentration into normal range. Now, note that this is the lower end concentration and this is the higher end. So what you need to do, because this concentration is higher still, we've got to bring it to this concentration at least. Now, where some students went wrong is they tried to reduce this concentration to the lower end, but what we need to do is just get it in to this range. So the calculation is 2.82 times by 10 to the minus 7 minus the 4.47 times by 10 to the minus 8. So this is telling us how much this concentration has to be reduced by to get it into this concentration. And because it's asking for a percentage, we need to divide that by the original concentration, so it's 2.82 times by 10 to the minus 7, multiply that by 100 to give us the percentage, and that comes out to be 84.1% change. Now again, remember, read the question. Okay, you've got a range here, so the trick with this was actually determining what value the concentration had to get to. So question 3, draw and label a single strand of DNA nucleotide. Now, the drawing for this is actually in the specification, and my advice is always go through the specification, and any place where it says the student should know this, so whether it be a definition or a diagram or some information, learn it, because they're going to come up in the exam. And it's easy marks, it's just things that you've just got to recall from the specification. Now, only 46% of students got this, showing that over half the students haven't learned parts of the spec where it just says you've got to know this. So the type of drawing that you want the ribose sugar, and to that we've got a base attached, and to this we have a phosphate. Now it says that we've got to label it, so we're going to label that as a phosphate. I'm going to label that as a base, and this is the deoxyribose. The second part, give two features of DNA and explain how each one is important in semi-conservative replication of DNA. Now again, a lot of students did the first bit, they give a feature, but they didn't explain how that's important, and that led to only just over 9% of students scoring the 2 marks for this question. So something that all students many students put down is there's 2 strands, but they didn't explain how that's important for semi-conservative replication. So the explanation is so they can be used as templates. So remember, when the DNA is being replicated, they split into two, and each strand becomes part of a new piece of DNA. So the feature is there's two strands. The explanation is so they both can be used as templates. So the second feature is there's weak hydrogen bonds between the bases. Again, that is giving the feature, but it's not explaining why that's important for the semi-conservative replication of DNA. So you've got to explain it. So this allows the two strands to separate. Okay, so we know that the two strands have got to separate, so they act as a template, and that can only happen because of these weak hydrogen bonds. So again, read the question, make sure that you're given the feature and the explanation. So the third part involves a calculation. And again, some students struggled with this. Now the question's saying that replication of mitochondrial DNA is different from nuclear DNA. The replication of the second strand of mitochondrial DNA only starts after 2/3 of the first strand has been copied. We've got a piece of mitochondrial DNA, 16,500 base pairs long, and it's replicated at a rate of 50 nucleotides per second. And we've got to show how long this entire bit of DNA takes to replicate. So we've been told before we can start replicating the second strand, we've got to do 2/3 of the first. So we know it's 16,500 long, and we've got to find what 2/3 of that is. So if we multiply that by 2/3, we get to 11,000. So it's got to replicate 11,000 of the first strand before it can go on to the second. So it replicates those 11,000 and then replicates the 16,500 of the second strand. So that comes out to be 27,500 base pairs. Now the time it's going to take is we've been told that it takes 50 nucleotides per second. So to find how many seconds there are, it's going to take, it's those 27,500 bases divided by the 50 nucleotides per second, and that comes out to be 550 seconds. So the answer is going to be C. So even students that got this right, the examiner's report said that many didn't show any workings at all. So the assumption is a lot of students who even got this right actually did it by guesswork. So question 4 gives us a picture of a part of a prokaryotic cell, and we're asked to name the structures. Now, one of the comments in the examiner's report is there was very poor spelling of some of these features. And also some students got a bit mixed up between membrane, cell wall and capsule. So I'll just go through quickly what each of these features are. So with W, which is this thin structure here, so that is the cell membrane. Then we've got a larger structure just outside the membrane, which is the cell wall. And then beyond the cell wall, we've got the capsule. And then these wispy protrusions from the cell, the flagellum. Okay, so then part 2 was name the main biological molecule in W and X. So W was the cell membrane and X was the cell wall. So W, the main constituent of cell membranes, are phospholipids. Now this next bit caught some students out. Remember, it's a prokaryotic cell. So what we're asked is, what's the main molecule in the cell wall? Now a lot of students put cellulose, but that's not true for a prokaryotic cell. So the correct answer for a prokaryotic cell is urea, or you could have had glycoproteins. Name the process by which prokaryotic cells divide. Again, you've got to have two parts to this answer. Fission is not good enough. It has to be binary fission to get the full mark. Now the next one involves a calculation. Okay, so we're told that these cells can divide every 30 minutes. And we've got a starting culture containing that many cells. So let's calculate how many cells there are after 3 hours. So we know that they multiply, they divide every 30 minutes. So first thing we've got to work out is how many times they're going to divide. So that's 3 hours divided by 30 minutes. So that's going to be 6 times. So 6 times from where we start, these things are going to divide. So we're going to start with a concentration of 1.35 times by 10 to the 4. Now remember, what they're going to do is double 6 times. So they're going to double, then double again, etc. Okay, so some students multiplied this value here by 6 or 24 or some other multiples of 6. But what we need to do is multiply it by 2 6 times. So that's 2 to the 6, which gives us an answer of 8.64 times by 10 to the 5. So question 5 is about triglycerides. So we're given a simple structure there and we're asked to identify what P and Q is. So P is glycerol, so we have a molecule of glycerol. And that is attached to 3 fatty acids. And the type of bond we get between the glycerol and the fatty acids are ester bonds. So next part is to describe how you test a liquid sample for the presence of a lipid and how you'd recognise a positive test. So you've been asked for what the test is and what a positive test would give you. So the test with this is to mix the sample with ethanol, then water. And it has to be in this order. If you mix it with water then ethanol, or water and ethanol, you're not going to get the mark. So this is where you've actually got to do it in order. So first, so you've got in your answer, you've got to make sure that you're saying you're first going to mix it with ethanol and then you're going to add the water. And a positive result, you'd get a white. So again, the colour is important to get the mark for this. You get a white emulsion. So question 6 shows us 4 different species of fruit fly. So it's telling us there's 4 species. So because we've got 4 of these smaller circles, we can assume that they are the species. So the question is, figure 5 shows a hierarchy. Explain how. So what we've got here, we've got circles within circles. We've got that one within a larger circle within a larger circle as well, and none of them overlap, so they're all distinct from each other, and we've got larger and larger circles encompassing more and more. So here we've just got one thing in this circle. This circle contains 2 things, and then this larger circle contains 4. So to explain how this contains a hierarchy, we just need to put that it shows smaller groups within larger groups, and there's no overlap between the groups. So name the taxon for which this belongs. So again, what we've got to know the difference between species, genus and family. So again, if we go up, so if this is our species, because we've been told there's 4 different species, then this is our genus, so therefore this must be our family. So the next part of the question, we're being told that these fruit flies display courtship behaviour. One of the stages of courtship is singing by males. Normally a male will produce a sign song in which a continual noise is made, and a pulse song which there is continual noise with some louder peaks. Scientists showed that fruit flies have a visual stimulus that made them sing. They made recordings of the song. So we've got, for the same time period, examples of 3 different songs. Okay, if you note that these last 2 are of the same species, but we have a male and a female. So we've got the sign part of the song and the pulse part of the song. So it says the 2 different species are closely related species, but they are different species. Describe evidence from figure 6 to support the statement. So because they're closely related, we're looking for some similarity. But because they're different species, we're again looking for a difference. So we're looking for something that's similar to show that they're related, close related, and we're looking for something that's different to show that they are distinct species. So we want to look at the male for both species, for the different species. So what we can see here is the sign song is very similar between the two. It'd be very difficult to look at that and determine which is which. So we've got a similarity, but the pulse song there's a definite difference. So here we've got the pulses that are quite uniform and quite close together, but here we've got the pulses are definitely further apart. So to answer this, we could put something along the lines of that we know that they're closely related as they are very similar sign songs, but we know that they're different species because their pulse songs are different. But again, it's not, because we're stating a difference, we've got to say how they're different. So we could say, but they have different intervals between the pulses of pulse song. So you could have also said they had different amplitudes, but you've got to state how the two species are different in terms of their pulse song, i.e. because of the intervals or because of the amplitudes. Okay, so the last part of this question, it says scientists repeated their experiments using female fruit flies as the visual stimulus. When the males and the females were together, their songs led to mating. When two females were together, their song did not lead to any attempt to mate. Use the information from figure 6 to suggest why two females did not attempt to mate. So it's telling us to look at figure 6. So we're going to have to compare, the male and the female songs to come up with an idea why they did not mate when you had two females. So if we look at these, if we look at the male and the females, so we can see that the peaks are at different times. So we've got, these peaks in the male, the first two are closer together, and then we've got the third. So in the female, we've got a long gap between the first and the second, and then we've got a shorter gap to the third. So we can see how the male and the female songs are different. So we need to explain that and why that difference wouldn't lead to mating. So we can say that with two females, the peaks would occur at the same time. But with males, would be at different times. Because of those gaps in between. Therefore, there'd be no male to stimulate mating. So remember, it's courtship that stimulates mating. So this is part of the courtship. You need these two different songs to stimulate the mating. So some students during the exam had put that one female or both females recognise that they're both females, so therefore they wouldn't mate. That's not what courtship and mating is about. You need a courtship ritual to stimulate the mating, and it's these two songs. The male song would stimulate the mating through courtship. So if that male song isn't there, then mating isn't stimulated through courtship. Question 7 asks us what a proteome of a cell is. This is another example of the definition for this is actually in the syllabus. So it's one of those things that's very easy to highlight as you go through the syllabus and learn it. And again, looking at the marks that students gain from this question, less than 18% of students got the mark. So it's another example of something that's there in the syllabus for you to learn. To be able to get the marks, you've just got to regurgitate the answer. So the answer for this is the proteome is the number of different proteins a cell is able to produce. Next part is give two structural differences between a molecule of messenger RNA and a molecule of tRNA. Now again, the key there is differences. So it isn't just enough to state a structural feature of one of them. You've got to say how it's different to the other. So for example, mRNA is linear, but that's not enough to get the mark. Yes, mRNA is linear, but you've got to say how it's different to tRNA. So you could say mRNA is linear, but tRNA is cloverleaf. So second feature we could have is messenger RNA does not have an amino acid binding site. And again, you've got to state what the difference is. But tRNA does. Okay, starting with mRNA in the cytoplasm. So again, it states that it's already in the cytoplasm. Describe how translation leads to the production of a polypeptide. So it's asking you for translation. It says do not include descriptions of transcription and splicing in your answer. Again, this is one of those examples I said right at the beginning of the paper where students just didn't follow the instructions given in the question. So many students wrote about transcription and splicing, and some talked about how the mRNA left the nucleus to go into the cytoplasm. We're given all this information in the question, Okay, so make sure you read the question, underline relevant bits, and follow the instructions. So once it's in the cytoplasm, we have the messenger RNA, and that associates with the ribosome. And then the ribosome moves to a start, and that is important. You've got to make sure that it moves to a start codon. So then we have the tRNA carries the appropriate amino acid, and the anticodon on the tRNA is complementary to the codon on the mRNA. Okay, so these two codons line up, the one from the tRNA with the one from the mRNA. So then the ribosome moves along to the next codon, and this process is repeated and the amino acids are joined by peptide bonds to form a polypeptide. So again, with this type of question, just go through in a logical order, describing each step in turn, but it's key to read that initial instruction to understand that you're not going to give any information that you're told not to. Question 8 is asking us to state and explain the property of water that can help to buffer changes in temperature. Okay, so the thing about water, because it's got a high specific heat capacity, then it takes quite a bit of energy to actually change the temperature. So if we just write that in a way that is concise, we can say the water has a high specific heat capacity so that will give us the mark for stating the property of water. So to get the mark for the explanation, therefore it takes a lot of heat energy to change temperature. So things will stay at a stable temperature unless there's a lot of heat energy input. Water is used to hydrolyze ATP. Name the two products of ATP hydrolysis. So with this, I think a few people got the base wrong. So it splits it into two. So we've got adenosine diphosphate, because what we're doing is we're removing one of the phosphates. So now it only has two phosphates attached to it rather than the three. So now we've got a table where we've got hydrolysis of ATP is catalyzed by the enzyme ATP hydrolase. The student investigated the effect of ATP concentration on the activity of ATP hydrolase. Okay, so we're changing the concentration of ATP and we're looking at the activity of ATP hydrolase. She used shortening strips of muscle tissue caused by the contraction as evidence of ATP was being hydrolyzed. So the shorter that the muscle strips became, the more ATP was hydrolyzed. She took 4 slides, A, B, C and D, and added strips of muscle tissue of the same length to each slide. She then added the same volume of ATP solutions of different concentrations to the 4 slides and left each slide for 5 minutes. She then recorded the final length of each strip of muscle tissue, and we've got the results there. So we can see that we've got different concentrations of ATP, and we've got the final length. It says, other than those given, okay, name 2 variables the student should have controlled. Now the reason I've underlined other than those given is because some students actually just give some of the variables that we've already stated in the question above to do with time, etc. So you've got to give 2 different variables that haven't already been given. So again, there is a problem with some of the answers that were given. So some students just put thickness. Okay, now whenever you've got a term like this, you've got to qualify by, what is it that you are controlling. So in this case, it might be the thickness of muscle tissue. You could also say the temperature of the muscle tissue. It's not good enough just to say terms like controlling the temperature or controlling the pH. You've got to say what it is you're controlling. Okay, you've got to qualify all of these terms. So only 7% of students who sat this exam got the 2 marks, mainly for those reasons, not actually given a qualified answer just by giving terms like temperature and pH. So here we've got to describe and explain the pattern shown by data in table 1. So description, that's all it is. It's just describe what's going on. So if we have a look at this, we can see that as the concentration of ATP is increasing, the actual length of the muscle is decreasing. So that's all we've got to do for this answer, is describe the trend. So as one's increasing, the other is decreasing. So that's all we need to put for the description part. So as conc of ATP increases, the length of muscle decreases. Okay, so now we've got to explain it. So as we're providing more ATP, then more ATP is being hydrolyzed by the ATP hydrolase. So then more energy is actually released. So there's more muscle contraction. Now, one of the things to really sort of spell home here is energy isn't produced, it's released. So again, students who put more energy would be produced, they're not going to get the mark for this because you don't produce energy, you're just releasing it. So the explanation we can put for this is more ATP is hydrolyzed by ATP hydrolase. So more energy is released. Therefore, there's more muscle contraction. Okay, so the last part of this question is a calculation. Now again, remember what I said right at the beginning of the paper about the math skills were commented on by the examiner. And this is a point in fact. So with this question, only 3.7% of students who sat the paper got the full 3 marks for this. So what we've got a concentration, okay, of solution and an amount of ATP releases this amount of energy. So we're told that 60% of the energy released during the hydrolysis of 1 mole per dm cubed of ATP is released as heat. So then the rest is used for muscle contraction. So i.e. 40% is going to be used for muscle contraction. So the student added this volume of ATP solution to slide D. Okay, calculate the energy available from ATP for contraction of the muscle on this slide. So I think many students just missed that slide D, so they ignored the concentration. Okay, so first thing we've got to do is work out how much energy could be released by the amount of ATP that we are adding. Okay, so we've been told that we are adding 0.05 cubic centimeters. So what we need to do is find what fraction of that is of a dm cubed, because the amount of energy that we've been told is released is based on adding a full dm cubed. So we're only adding a fraction of that. So the fraction of a dm cubed is 0.05 over 1,000. So that's the fraction of a dm cubed that we're given. Now we've been told that this amount of energy will be released based on this concentration of ATP solution, but we've been told that we're using slide D. So if we go and have a look at slide D in the table, we've been told that slide D is 8 times by 10 to the minus 6 moles per dm cubed. So we've worked out the fraction of the volume added compared to the 1 dm cubed. And now we've got to multiply that by the actual concentration of the ATP that we're using. So in this case, it's 8 times by 10 to the minus 6. So by having the volume that we're adding and the concentration, what this calculation would tell us would be the amount of ATP in moles that we're adding, okay, which is obviously a fraction of what this is. So we've calculated the fraction compared to 1 dm cubed of 1 mole per dm cubed. Okay, so this fraction we just calculated will be the fraction of this energy that we released. So if we multiply this fraction by the 30,500, that will tell us how much energy is going to be released by this volume and this concentration of ATP. And that comes out to be 1.22 times by 10 to the minus 5 joules. Okay. We've also been told that only 40% of this will go for muscle contraction. So we've got to work out what 40% of this is. So it'll be 40 divided by 100 times by the 1.22 times by 10 to the minus 5 gives us 4.88 times by 10 to the minus 6 joules. So that will be the amount of energy that's been released that will be available for muscle contraction. So it's a two-step process, this question. We've got to work out how much ATP is actually being added by knowing the volume and the concentration. And then, based on that amount of ATP, work out how much energy is going to be released. And because we've been told there's only a certain percentage of this energy that's being released can go for muscle contraction, that's how we get this final answer. Question 9. We're asked to describe a pathway taken by an oxygen molecule from the alveolus to the blood. So 70% of students scored 0 for this because they gave a mechanism rather than a pathway. So the pathway is just that the oxygen passes across the alveolar epithelium and then the epithelium of the capillary. Explain how one feature of the alveolus allows efficient gas exchange to occur. So I've underlined alveolus there because it's asking us for a feature of an alveolus, not the alveoli. Okay, so a lot of students talked about large surface area. Now that's a consequence of the alveoli. So this is an individual alveolus, which is to do with it being a very small diffusion pathway. So the answer to this will go along the lines of the alveolar epithelium is 1 cell thick. For 1 mark, which creates a short diffusion pathway. So again, it's important to read the question and see exactly what it's asking you for a feature of. Okay, so we've got some data in the form of a table here. So what I'm going to do is just go through some of the important features of the table. So we've got carbon monoxide as a poisonous gas, which is present in cigarette smoke. So it's telling us that it's poisonous. Okay. The carbon monoxide can be absorbed into the blood where it binds to hemoglobin. Scientists investigate the concentration of carbon monoxide in cars in which people were smoking or not smoking. They measure the concentration with the car windows open and closed. Figure 7 shows scientists' results as they presented them. A value of plus or minus 2 SDs from the mean value includes over 95% of the data. So the fact that it's specifically telling us about these 2 SDs and it's got it on the, so if we look at the key, we've got that this bar shows plus or minus 2 SDs. We're going to have to include this in our answer. Now where the 2 SDs come into play is talking about whether differences are significant or not. So for example, if we look at this, these data here and these data here, there is a difference, but because there's an overlap of the 2 SD, so we can see that that gives us an overlap over here. We can't say that this difference is significant, but we can say the difference from here to here is a significant difference because there's no overlap of the SD bars. So if we look at what's going on, we can see that smoking after 5 minutes, again, if we go down to the key, with the closed windows, there is a definite difference, even after 5 minutes, between the carbon monoxide levels for the non-smoking and the smoking. Okay, but if we look at when the windows are closed, which is given by the white ones, the value for the smoking with the windows open and the value for the non-smoking with the windows open, actually there's not a significant difference because we do have an overlap with these SD bars, and same over here. So with all the data with the open window, we can't say there's any significant differences because we've got the overlap of the S2 bars. But we can say there's significant differences with the data when the windows are closed because we don't have an overlap with the 2 SD bars. So if we have a look at the question, it said that the law was changed in 2015. So as following the introduction of the law, politicians stated it's dangerous to smoke when a child is in the car. Higher levels of deadly, so I'm going to underline that, toxins can build up even on short journeys. So even on short journeys, and children breathe faster than adults, meaning that they inhale more of the deadly toxins. Use the information provided and the data in figure 7 to evaluate this politician's statement. Okay, now if we look at the marks that students gained during the exam, just over 1% of students got all 4 marks because not enough students were making use of the data that was in the bar chart, or making use of that term significant difference, i.e. pointing to the overlap or the non-overlap of the two SD bars. So the fact that in the question it's pointing you towards these two SD bars, it wants you to refer to them in the answer, and that implies whether the data is significantly different or not. Okay. The other things that students did during this is many repeated themselves. So rather than give a concise direct answer, they just repeated themselves all through the question. So now we've had a look at those tables, let's start putting the information in the answer. So it's asking us to evaluate this statement, which implies that we're going to have to give some responses that back up the politician's claim and some responses that refute, that are against what the politician's saying. So some of the things that we agree with this politician statement is there is a significant, again, use of these terms, significant difference. So there is a significant, and here it's important to say what that difference is. So whether it's higher or whether it's lower, rather than just be vague with there's a difference. So there's a significant higher concentrations of CO compared with the no smoking. So again, because it's a difference, we've got to say what we're comparing with. So it's a significant higher concentrations of carbon monoxide compared with the no smoking. As there's with the windows closed. And explain why we think it's significant, as there's no overlap in 2SD. Okay. So we've talking about, so the other thing that the politician says up here is that it's dangerous to smoke when a child's in the car. Well, that's true, because we know that any increase in carbon monoxide could be dangerous, because we've been told that it can bind to hemoglobin, so it's going to have less oxygen to be carried around the body. So again, this is another thing backing up what the politicians said. We can say that any increase in CO could be dangerous. As it causes less O2 to be carried. Okay, so we've given 2 points that we can confirm what the politician's saying. The politicians also said that the deadly toxin, or the toxins, can be built up even on short journeys. Now we saw that there was that significant higher level just after 5 minutes. So that backs up the comment about short journeys. So we can also say that there's significant higher levels after 5 minutes with the windows closed, supporting short journey statement. Okay, so now we could put some arguments that are against what he said. So we've already talked about that there's no, so if I just put against. So we've said there's actually no significance when the windows are closed, when the windows are open, because there's no 2D, because there is a 2D SD overlap. So we can say that there's no significant difference with open windows as there is a 2SD overlap. And we can also, so if we go back to the table, the types of concentrations we're talking about are between 4 and 5 parts per million. Okay, now in the statement he said they're dangerous, and they are dangerous because we know what carbon monoxide does, but he said that they're deadly toxins. Now, there's no evidence in what we've been told, what we've been given, where the 5 parts per million is actually a deadly level. So we can't say for sure that what he's saying here is actually true. So again, with the against, we can say that there's no idea given in the statement if 5 parts per million is deadly. We could also comment on the fact that he's put this thing about children breathe faster than adults. So again, we're not given any evidence in this question about whether children do breathe faster. We know that children's lungs are smaller, but we haven't been given anything about breathing rates. So again, we could comment that this statement by the politician about children breathing faster, we can't actually verify that or not. So question 10 is a comprehension question. So we're going to have to make use of some of the information that we're actually given in this passage. So what I'm going to do is just go through and underline a few things that pop out. So it says sizes of populations in normal intestinal bacteria are usually controlled by T cells. So we've got normal bacteria and T cells, and they're produced slowly and in small numbers by the immune system. These T cells do not normally survive very long. Okay, so not survive very long. As a result, they do not release large amounts of cytokines, which are chemicals that cause swelling in the lining of the intestines. Crohn's disease is a long-lasting disease that causes swelling of the lining of the intestines. So we've already got a link there. So we know that cytokines cause swelling, and we know that Crohn's disease has this swelling of the linings of the intestine. It's believed that Crohn's disease can be caused by a loss of tolerance to normal intestinal bacteria. Okay, as shown by unusually large response to T cells. This response can be triggered by pathogenic bacteria in the intestines of people with a genetic tendency to Crohn's disease. Some people with Crohn's disease can be controlled by a drug, this 5-ASA, that reduces swelling. So again, the swelling is mentioned again. Another drug, 6-MP, may also be used. 6-MP inhibits an enzyme required to make A and G. This is effective because most cells can recycle nucleotides, but T cells cannot recycle these bases. Use the information from the passage and your knowledge to answer the question. So again, we're going to have to refer to some information that we've just been given. So it says the Crohn's disease symptom of swelling in the lining of the intestines can be triggered by pathogenic bacteria in the intestines. Suggest how. So again, if we go to the relevant passage, it says that Crohn's disease is a long-lasting disease that causes swelling. It's believed that Crohn's disease can be caused by a loss of tolerance to normal intestinal bacteria. Okay, so if we go back to the question, so it says it can be triggered by pathogenic bacteria, but we've also got this thing about normal bacteria in the gut as well. So we've got to link and see how that's going to cause the swelling of the lining. And we know the swelling of the lining is to do with the cytokines. So we can say the presence of the antigen on the pathogenic bacteria because we've got to suggest how it's triggered by the pathogenic bacteria, causes more T cells to be produced. So it's the presence of these pathogenic bacteria that causes the T cells to be produced. And the T cells are then going to act against the pathogenic and the normal bacteria. So these act against the pathogenic and the normal bacteria. So what's going on here, how the Crohn's symptoms disease of the swelling of the lining could be triggered by pathogenic. So the pathogenic bacteria makes more of these T cells to be produced, and the T cells act against not only the pathogenic but the normal bacteria. We could also, they're all 3 points, but we could also go on and rather than some of the points we've already made, talk about the fact that the T cells are longer lasting, and so therefore more cytokines are made, causing the swelling. Okay, so again we can go through this by saying that because more T cells are produced, being longer lasting, they're going to produce more cytokines and therefore more swelling. Suggest the meaning of a genetic tendency in Crohn's disease. So we've got to be quite specific here and use, as I've mentioned before, specific language because we're doing A-level. So terms like people passing it on are just not good enough. We're not going to get the marks. So when people have a genetic tendency, we're talking about a mutation or an allele. So let's start there. So we can say that some people have an allele that increases the chance of them having an unusually large T cell response. Okay, so we're keeping it concise and we're using scientific language like alleles. Okay, so when we said suggest a meaning for the genetic tendency is passed on, but because they have certain alleles that and we're linking it to the Crohn's disease, so we're linking it to the fact that they're going to have an unusually large T cell response, which we've got from the passage. So again, keep referring to the passage. So we've used some knowledge that we've already had, that we've got about alleles, linking that to a genetic tendency, and we've got some information that we've gleaned from the passage, i.e. the Crohn's disease is characterized by having an unusually large T cell response. Suggest why 5-ASA is only effective in controlling the swelling of the lining of the intestines of some people with Crohn's disease. So in the exam, some people talked about people being allergic, digesting the drug, or having an immune response to it. But there's a couple of reasons why this drug might not be appropriate to some people. So some people may be allergic to 5-ASA. Like a lot of drugs, people can be allergic to them. So therefore, and again, we've got to explain why, then really hit the examiner over the head about why I can't take it. So yeah, you've said people might be allergic to it, and that's implying something, but you've actually got to say, so therefore they can't take it. Because the question's saying, suggest why it's only effective in some people. Well, the reason that it's not effective in these people is because they can't actually take it. Okay, so we could have also had that some people might produce really large amounts of the cytokine, and therefore the 5-ASA are not going to be good enough to reduce that amount. So the last part of the question suggests why 6-MP can be used to control the symptoms of Crohn's disease. Do not include details of enzyme inhibition or protein synthesis. And again, this is an instruction that was ignored by some students, and some students talked about enzyme substrate complexes despite being told not to. So if we go back to the passage and see what it says about this drug. So it says that 6-MP inhibits the enzyme required to make A and G. Okay, so this enzyme means that we can't make this. This is effective because most cells can recycle nucleotides, but T cells are not able to do so. So what it's saying is, in a lot of cells, even though we're inhibiting the ability to make A and G, in T cells it's more significant because they can't recycle it. So again, we're going to have to use some of this question is saying that showing that we can extract information out of these passages to answer a question we've been given. And again, we're not to talk about enzyme inhibition. Okay, so if this cell doesn't, or the T cells don't have A and G, then what does that mean? So we can say that a lack of A and G will slow DNA synthesis in T cells. Okay, so what we're doing was we are linking the lack of A and G with what it's going to do to the cell. So it's going to reduce the rate that DNA can be synthesized. And so we can say that it's only going to affect the T cells because they can't recycle it. Okay, so we can say that it affects T cells more, because it wouldn't be a very good drug if basically it was doing this to every single cell in the body. You're going to have massive side effects. So it's only going to affect T cells more, as they can't recycle these nucleotides. Therefore, 6-MP causes fewer T cells to be produced. And we know about the T cells and the cytokines, so this causes less cytokine to be produced, therefore less swelling. So what we've combined some of our knowledge of biology, i.e. a lack of DNA bases is going to slow down synthesis, and combine that with some of the information that we've been given in the passageway. And that's the point of these comprehension type questions, being able to show that you can extract the relevant information out of the passageways that the question is asking for.